Good morning, friends. I am Dr. Sampada Patharkar, Assistant Professor, Department of Botany, from Rajeshri Shahu Science College, Chandur Railway, Amravati University, Maharashtra. Today, I will show you practical demonstration on double staining and making permanent slide. Double staining means staining the material with two different stains. Usually in double staining, dear student, saffronin and light green are used. Instead of light green, we can also use aniline blue and or the fast green. Saffronin stain with thick wall lignified cells such as xylem, sclerenchyma, etc. Whereas the light green stain with thin wall living cells like sclerenchyma and collenchyma. Now we are moved towards the live demonstration of how to make the double staining permanent slides. The material you require for these experiments or what we have here on these tables. Plant material for section cutting. This is a stem of Dracaena plants. One razor blade for section cutting, for fine section cutting. One brush. One cavity block with water. Second one, saffronin solution. Another one is cavity block with water. Then 30% alcohol, 50% alcohol, 70% alcohol, 90% alcohol, absolute alcohol, fast green, xylene, DPX, 2 needle, cover slip, slides, and last one is microscope for observing your fine section. First of all, you have to hold the plant material in your left hand with your two fingers, that is the thumb and index finger. Use your middle finger to support to index finger. You have to keep the ring finger just below the material. This ring finger will help you just taking the material upward. Whenever you taking the section, hold the razor in your right hand in this way. Move the blade over the material from one end to the another. Now you have the section over the blade. Like that, you have to take a series of section. When like this, now all the sections are on blade. Now keep the blade on water and shake it gently, and all the sections are come into water now. Select the thin section out of these with the help of brush and transfer it into the saffronin solution. Before select the thin section, one thing kept in your mind, that is you have select the entire section for further procedure of making the permanent light. Okay, now I will transfer the section into the saffronin. In saffronin solution, you have to keep the section in 3 to 5 minutes. So, during this process, one thing raise in your mind. What do we use the preserving material? Dear student, the material is usually preserved in this type of the bottle. For preserving material, you can use the acetic alcohol and 4% formalin acetic alcohol. Means it is a mixture of acetic acid and alcohol in 1:3 ratio. Now take the section from saffronin and transfer into water and wash this section very gently. After completing the washing procedure, the material transfer into 30% alcohol. In 30% alcohol, the section in 3 to 5 minutes. When we dissolve the saffronin in 30% alcohol, you can take the section directly from saffronin to 50% alcohol. After 3 to 5 minutes, remove the section from 30% alcohol and transfer it into the 50% alcohol. For 3 to 5 minutes. After finish the timing of 50% alcohol, you keep this section out of it. That is the 50% alcohol, and move into the 70% alcohol. You have to keep this section in again 3 to 5 minutes also. Dear student, why we use the alcohol grade? 30% alcohol, 50% alcohol, 70, 90, absolute alcohol. Because alcohol grade is used to remove the water from section. This process is called as dehydration. When the sections are totally free from water, there will not be any bacterial or fungal contamination. After that time, you take the section out of it and transfer it into the 90% alcohol. In 90% alcohol, now the section in absolute alcohol for 3 to 5 minutes. Dear student, why we use so many alcohols, 30% to absolute alcohols? Why cannot we use directly the 100% alcohol grade from saffronin to the absolute alcohol? Because if we take the section from saffronin to directly 100% alcohol, the section will shrink and all the cells will lose their shape. So we cannot see the cells in proper order. Dear student, remember this thing. Always, you have to keep all the alcohol grades in this manner. Whenever you're doing all this. Now, I will tell you again one question. That means what will happen if we don't cover the alcohol grades? Because alcohol evaporates faster than water. So the alcohol evaporate percentage will be reduced. For example, 50% alcohol may become 40%, 30% alcohol may become 20%, and so on. So now take the section from absolute alcohol and transfer it into the fast green. In light green solution, you have to keep the section in half to 1 minute. After timing, take this section from the light green with the help of brush and transfer it into the xylene for 1 to 2 minutes. We using xylene solution to clarify the specimen in the last step before it is mounting. It removes the excess of solution. That's why we used here the xylene solution. Now you take the slide. Okay. I will take this slide and wipe it with very gently. Now this is very clear and neat slide for taking DPX and section. First of all, I will take a DPX mountant solution with dropper. With a dropper, I will put on DPX solution on a slide very carefully. Now you take the section with needle and kept on over the slide. Okay. You have to take the cover slip and put it over that with the help of needle. Okay, you have to see that there should not be any air bubble. Any air bubble. Now you have the double stain permanent slide. These can last for 5 to 6 years. So now you take the section and observe under the microscope. I will take the microscope. And observe. under the microscope, that time the section look like this. It is very colorful because double staining procedure. Staining is essential. Why? Because different tissues and other structures can be easily differentiated by staining. But members of pteridophyta, gymnosperms and angiosperms are stained by the double staining method due to presence of differentiation of tissues. I hope this video upgrade your knowledge regarding double staining. Thank you.